So I'm going to be talking about not venom for the first few slides, because there's a few other concepts I want to introduce to everyone, because not everyone might be familiar with them. But I will get back to venom, don't worry about it. So really the key point of this, which actually follows on quite nicely from the previous monotremes talk, is that species richness varies hugely across the tree of life. So this is just a tiny, tiny fraction of the number of beetle species. This is the entire diversity, modern diversity of monotremes. So you can see why those two things differ. And a large part of evolutionary biology research over the last 150 years, more or less, has been trying to understand why there is this huge variation. And when we think of species richness, really that's a function of two things. First is time. So in this little toy example, we have 6 clades within this larger group, which have different number of species contained within them. As you can see, older groups, which diverged earlier, tend to have larger number of species. So if you plotted species richness against clade age, older groups, more species, because they've had more time to build up those species, accumulate them over time. Alternatively, in this dummy example, you can basically see in actual fact the opposite, but nevertheless you've got old clades with very few species and relatively recent clades with a lot of species. So it's not the case that old lineages are more diverse, but these younger lineages have diversified much faster. So if you plotted species richness against diversification rate, you would see this positive relationship. So really diverse groups are those which have speciated very, very quickly and diversified rapidly, because they can generate more species in a given unit of time, so in each million years, for example. Now, these two things always play a role, but they play a different amount of role. So the amount of variation in species richness they explain varies massively across different groups of animals and plants. And of course, because we like to go for the big showy extreme scenarios, when we're thinking about explaining diversity and why some groups of animals are so species rich, we tend to turn to these super radiations of fishes and insects. If we imagine this black square, the area of this black square is the entire diversity of vertebrates, and this one is the entire diversity of invertebrates, then you can get a handle for just how species rich these two subgroups in those are. So everything in white here is not an insect, and you can see that leaves out of the about 1 million species of invertebrates, 75% of them are insects alone. It's slightly less showy for fishes, but out of vertebrates, about 48% of those species are fishes, which means this might be just more than half, but that includes every mammal, every bird, every amphibian, and every reptile that you could think of is contained within here. Collectively about the same diversity as fishes alone. So there's not much you can do about age, not much changes age, time just wanders on as it always does. But we know that traits that organisms possess can affect the diversification rates, and therefore we can have situations where traits possessed by organisms or lineages can make them speciate faster or slower or whatever else. And thinking about insects specifically, traits like herbivory have been proposed to do this previously. So this is largely because herbivorous insects tend to be involved in evolutionary arms races, kind of chemical warfare with the plants that they're feeding on. That spurns diversification. Metamorphosis kind of disconnects juvenile and adult life stages and allows a broader range of selection pressures to be at play, and that's been suggested to influence it. And the evolution of wings in insects has been proposed to increase diversification. Now, actually, some of the results I don't have time to speak about today, but came from the same paper, suggest that there's issues with all three of these, or at least the evidence for all three of these. I can speak more about that if you're interested. But in terms of fishes, it's mostly habitat that seems to be important, given previous research. So those that live in coral reefs or those that live in freshwater habitats tend to be faster speciators, so higher diversification. How do we explain the mega diversity of insects and fishes? Well, we could consider clade age. I mean, both of these groups have extensive fossil records, and they are both over 400 million years old. That's a lot of time to accumulate species. We also know there's examples from both fishes and insects of rapid radiations. So it could be that diversification rates are higher in these groups, and that could be really important. I told you to get back to venom. Or it could be that venom is a trait which mediates diversification rates. Now that seems a bit odd just thrown in there, so I just want to explain briefly why we think that venom might be important in this way. The first is that, as I mentioned earlier with herbivorous insects, antagonistic coevolution favours diversification through arms races. That's been widely assumed, and there's some evidence for this going back several decades. We know that venom has several functions, from predation, defence, interspecific competition, etc. But all of these are involved in antagonistic coevolutionary interactions. So we think that these should lead to diversification via this mechanism. Previous work by myself and Richard Harris, who was also involved in this project, and Mike Speed earlier on, has basically showed that venom is associated with faster diversification in tetrapods, but not in amphibian toxic defences, by which mostly mean poisons. So it could be that these relationships that were found before are specific to amniotes because they weren't really found in amphibians, or they could be specific to venoms because most amphibian toxins are poisons rather than venoms. So I wanted to broaden that out and also ask whether we can explain some of the diversity of insects and fishes. I'm not going to go into detail on any methods, I can do that happily later on, but essentially we took a family level approach using evolutionary trees and extracted data on species richness and the presence of venom in each family from the literature. Calculated diversification rates using this method. A whole range of different analysis on them. One type, which is similar to a regression, which you might be more familiar with, I'll focus on in this talk. And we asked 3 questions. Firstly, are diversification rates the most important determinant of species richness in insect and fish families? Because if it's mostly to do with age, then venom doesn't have the opportunity to have much effect. Is venom associated with higher diversification rates? So do we find this pattern again? And if we're thinking that venom may have played a role in generating the diversity of fishes and insects, there's no point in that just being a tiny little proportion of the history. So it has to be over quite a large proportion of the evolutionary history for that to be important. So we tested these 3 questions. I'm not going to get you to read this whole table, but the key thing here is that diversification rates typically explain far more variation in species richness than clade age did in both fishes and insects. So that's good. There was an additional effect of venom that was significant, although relatively small. This could be an issue because it could suggest that we're finding this result simply because venom is more likely to evolve in bigger families because there's more species in which. But this extra effect over and above diversification rate in clade was actually really small, less than 1 species in both groups. And it's not related to age. So if it's related to anything, it must be diversification rate, and that's sort of what we're interested in. The key result is that, yes, venomous families of both fishes and insects diversify much faster than non-venomous families, in fact, about 2-fold faster. So we do have this effect where venom is associated with really rapid speciation, or at least diversification rates over time. Using ancestral state estimations, we managed to find that in fishes, venom has evolved a minimum of 19 times and been lost in over 11. And in insects, we find 28 origins at a minimum of venom in insects with up to 2 losses. This is just the family level. More than that if there's been gains and losses within families. And if you take all the total evolutionary times, you add up all these branch lengths, which represent time, it turns out that venom has been present for about 10% of the total evolutionary history of fishes and about 14% that of insects. So it's really quite a large proportion of their time has been spent in this state, which doubles their diversification rate. So there's plenty of opportunity for this to happen. What's sort of interesting looking at the patterns of origins across here is that insects are basically, venom has evolved at most time points pretty much throughout their history. However, in fishes, you tend to find that the origin of venom in fishes is mostly, the vast majority of those origins are separated into 2 fairly narrow time windows, which correspond to the late Cretaceous and the Eocene periods. Okay? Now, what happened in those periods? Well, we know that fish venoms are almost entirely defensive, so we know it's likely to be some kind of change in the predation landscape that's resulted in this kind of burst in families of speciation, or venom evolution, sorry. And well, what's happening in the late Cretaceous? Well, pretty much every other large marine group, predator group, was declining in the late Cretaceous. Think of things like ichthyosaurs. However, mosasaurs actually originated and peaked in diversity during this period. So this new group of predators that was really quite ferocious came in just at the time that all these origins of venom will happen. Similarly, in the Eocene, this is basically when cetaceans, so whales and dolphins, originated, and they also peaked in diversity around this same time. And this is after the end Cretaceous mass extinction, so this is after things like mosasaurs and other previous large marine predators. Now, why would these two groups potentially lead to the origin of venom? So basically similar evolutionary effects on their prey. Well, it turns out that from the best paleobiological information that we have, mosasaurs are basically thought to have been a lot like cetaceans. You can see that the broad body form was fairly similar, but they are also thought to have been very active predators, and mosasaurs are also thought to be endothermic as well. So you've got very similar movement and potentially very similar impacts on the prey populations. So we sort of cautiously speculate from this because we don't have fossil evidence of this, but based on the similarity in the ecologies of these two groups and their coincident times of the origin and peak of diversity with the two time windows of venom evolution in fishes, that most origins of fish venom have been linked to predatory pressures that were very similar in the late Cretaceous by mosasaurs and in the Eocene by cetaceans. I think that's really cool. So briefly to summarize, the diversification rate variation is an important determinant of species richness in both insects and fishes. Venom is associated with several-fold, at least two-fold higher diversification rates in both groups. And it's evolved many times in both insects and fishes. And a substantial proportion of the evolutionary history has been spent in possession of venom. In other words, our results provide evidence that venom has played a part, not necessarily the major role, but certainly a substantial part in generating huge diversity in the largest vertebrate and invertebrate animal radiations. So with that, that's perfect timing. I'm just going to stop now, and I'm happy to speak further in this.